In this video, we will discuss about molecular mechanisms that involve FOXP3 regulation in T cells using CRISPR screening. So why the researchers decided to find out these FOXP3 regulations in the T cells? Because FOXP3 is been known to play a role towards immunological response or immunological tolerance in the T cells. That's why they wanted to do systematic studies to find out what are the uh, genes that cause these upregulations of this FOXP3. Although FOXP3 can be triggered using TGF-beta in the activated T cells, but they wanted to find out the genes that are responsible for this FOXP3 upregulations. So how they did their studies? They took the primary T cells, that is CD4 plus T cells from the patients, and then transduced these guide RNAs libraries using lentiviral pool, and then electroporated Cas9 proteins. Thereafter, edited cells were then used using the FAC or flow cytometry, or this flow sorter using flow cytometry to differentiate between the cells that are FOXP3 that can express FOXP3 or that does not, that has low expression of FOXP3. So before that, they activated the T cells using this cell surface antigens using anti-CD28 or anti-CD3, and also they have kept these cells under TGF-beta for some times, around 72 hours. Thereafter, FOXP3 has been activated, and they sort the cells between FOXP3 positive, that is, that shows the top 15% of the FOXP3 high, and it shows the top less bottom 15% of the FOXP3 low. And thereafter, they use the transcriptomes analysis to find out what are the genes that are upregulated and downregulated in this case of FOXP3 high population and FOXP3 low populations. And most of these genes, they are able to most of the genes that is known to upregulate this has a positive impact on the FOXP3 upregulations, and the negative impact of FOXP3 downregulations are being identified in these studies. At the same time, they are also able to identify some of the novel or some of the new genes that also has a role towards the FOXP3 upregulations or downregulations, such as this SMAT3 or this SMAR CB1 has a role towards FOXP3 downregulations, whereas this SIK3 has a role towards FOXP3 upregulations. Thereafter, they also use gene ontology to find out what are the pathways that are activated in the FOXP3 negative regulators that acts as FOXP3 negative regulators, and pathways that cause that acts as FOXP3 positive regulators. So one of the top pathways is intracellular notch that regulates the transcriptions, and one of the top pathways that cause this, that is related to the positive regulations of this FOXP3, is the transcription complex assembly or the chromatin organizations, histone modifications. These are the thing occurs where in the FOXP3 positive regulator sites, or that upregulations of these pathways will promote FOXP3 upregulations. And then thereafter, what they did is they check these cells for they did the individual knockout of some of these genes that are found to be upregulated and downregulated in the patient's T cells, and check whether it will has the same impact that they have observed in the pool of CRISPR screen libraries, and they are able to see the in, in general, they are able to find out that the genes that are known to be negative regulators in the CRISPR screening pool has low FOXP3 expression, and the genes that has high, that is positive regulators using the genes that are identified as the positive regulators is known to have high FOXP3 expressions. this is what they have find out, and they did it for the negative. They did it for this FOXP3 negative regulators, and also and this in the panel. So panel E and F are shows the quantitative analysis or the analysis that has been done multiple times, whereas this panel G shows one of the representative of the flow cytometry results, where we can see that in one of the cases they see upregulations. The first column of the panel G shows the upregulations of this FOXP3, whereas second column shows the downregulation of the FOXP3. So these things has been done multiple times for the genes that are mentioned here, to show that the results from the CRISPR screen is more or less seems to be true, as they did the individual knockout libraries and then analyze. They did the individual knockout of the genes in the T cells and then check the FOXP3 expressions in those cells. So this is how they made the CRISPR screen pool, and they validate the results from the CRISPR screen pools is more or less same that they have observed from the single gene knockout. So we will discuss further results that are being shown here about the FOXP3 expressions, but for now they have shown their authenticity of their CRISPR screen pool that they have generated, and their results when compare it by comparing it with the individual knockout in the primary T cells. That's all for this video. If you need to understand more about this experiment or how they made their CRISPR screen libraries, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.